So we should probably welcome back Olivia. She made it back. She's had a rough semester so far, but she's back with us. And then we also have a couple prospective students that are also visiting with us. Is it Una and what was it? Angelina? All right, so welcome to you as well. So the plan for today. I've been kind of going back and forth on what to do about the NEUR worksheets. Some years I've basically like read through the whole worksheet, and that gets really, really dry and really boring really fast. Other times I've kind of plotted out time where we could kind of go over it, and I don't know. Again, it hasn't really seemed very productive. So here's what I decided to do. I just posted an answer key. So that went up. You may have seen me do it like 2 minutes ago. So my suggestion is, I do want to go around and collect these because they are due today. I need to confirm that folks did them. But I would ask that you compare your answers to the worksheet or the answer key that I just posted on Canvas. And then what we could try to do is, Monday or perhaps even Wednesday, we could, if there are questions, I'll sort of just leave some time at the beginning of each of those classes. And we can go over anything that you'd like to go over. Sometimes people have wanted to go over labeling of different structures or classification of different structures into like the different primary vesicles or secondary vesicles of brain development, that kind of thing. But for now, what I would like to do is just, I would just like to collect them. So if you could just pass them over, make sure your name's on them somewhere. I will get them back to you on Monday. We'll go from there. Thank you. Thank you. Thanks. Steal that from you. Thank you. Thanks. I already grabbed yours. I'll come back. Awesome. Including, wow. You guys are coordinated. Can I steal it? Thank you, thank you. All right, so like I said, I'll get these back to you on Monday, and I'm just checking to make sure people did them. Okay, as far as next week goes, so we have 1 week until break. So the plan is, as far as things coming up in this coming week, I think the big thing is there'll be a quiz probably, I'm saying midweek, that's probably going to be due more towards the end of the week on Chapter 7. And that'll needs to be due after we've had time to talk about it. So probably we're looking at maybe Friday. There'll be a quiz that'll be due. I'll try to get it posted early in the week, though. Also, your paper summaries are due on Friday, March 1st before you go off to break. So please post those. I'm happy to screen articles for you. Several people have sent them to me. I think I've responded to most of them. If I haven't yet, it's because I got behind. Oh, good. Come on, have a seat wherever there's an open spot. If I haven't responded to you, maybe send me a follow-up because I've just gotten crushed this week with emails and I've gotten a little behind on some things. It helps me, especially if you send the thing to me over a weekend or if I'm at home in the evening, if you can send me the PDF as opposed to a link, that is more reliably something I can actually get to and view, whereas the links kind of depend on the journal. If I'm off campus, sometimes I don't have access. So send me the PDF. I'm happy to like sort of glance at the article, make sure that it seems appropriate. The key things I'm always looking for, though, just so you know, I mean, I'm not going to read your article per se, but I'm just making sure it needs to be primary literature, so it's not a review. There needs to be a study that's being presented for the first time in that paper. It needs to have some molecular methods. I haven't really been checking the dates reliably, but you should be able to check the dates on the publication. But those are the big things. So primary literature, molecular methods, some kind of a scientific journal article. If you're having any trouble, do check the library primer that I posted that's sort of, alongside the handout on the assignment. Yeah. Yeah, a correlational study could be fine. So, one where they're just basically taking some people and measuring something. Well, I don't know. That's not true, is it? Bless you. I'm going to retract what I just said because I think one of the stipulations I did want it to be a manipulative experiment. And so if it's manipulative, it really is more of a causal type of a study. So I think it should be manipulative. So they're taking a group and manipulating something. And once you do that, then I feel like we can start to say it's a causal study. Other questions? All right, so that's basically our main plan of attack. I need to get your homework assignment for this chapter up too. So we'll probably have that due sometime midweek as well. Aside from that, what I'd like to do today is focus on continuing on with this somatosensory chapter. So here's a good place to start. I'm going to do a quick recap. So we were talking about different kinds of mechanoreceptors. And so this is where we've been, I feel like, for a couple days. So this slide was sort of overviewing different varieties of sensory receptors that we use to detect different kinds of touch. It could be light touch, it could be more of a pressure type of a sensitivity. And we talked about the size of the receptive fields. Some of them are really fine discrimination types of things. So each receptor has a very small little receptive field, and we gain a lot of resolution in terms of figuring out, where things are on our skin and what the shape of things are that we're clutching and touching by comparing the information coming from each individual little receptive field. So we can compare the input coming in from each of these different receptive fields for Meissner's corpuscles and get an idea of what the texture and the shape of the object is that we're looking at. Whereas Pacinian corpuscles that have a much larger receptive field, we don't get that kind of resolution. We just kind of know is something on our finger or not, based on that information. And then we also talked about the fact that some of them acclimate really quickly, or in neuroscience they say they call it adapting really quickly. I never really liked that because I'm an evolutionary biologist. So it's not adaptation in terms of like change in gene frequencies in your population over time. It's adaptation in sort of like an individual level. So this term adaptation, this just basically means that your sensory system is adapting to the stimulus. It's basically ceasing to respond to the stimulus after some time. I call that acclimation. That would be my preferred term for it. But that's not where the field is. But in any way, some of the sensory receptors we talked about, like Meissner's corpuscles and Pacinian corpuscles, they rapidly adapt. So they respond really quickly when the stimulus first appears, but then they kind of stop paying attention to it very quickly. Whereas others, like Merkel discs and Ruffini endings, tend to be much slower to respond. So, that's not really the right way to put it. They respond once the signal appears, but they never really stop responding. They just sort of keep in this sort of tonic manner. They keep responding over and over and over. So they're very slow to adapt if they ever really adapt at all. So like Ruffini's endings here, you can see this is indicating there's almost no change in the action potential frequency from the onset of the stimulus to the time that they finally remove the stimulus here. It's pretty close to constant. It's a little faster here, but it's really close to constant. So there's very minimal adaptation. We call these slow to adapt ones tonic receptors, whereas we call these rapid to adapt ones phasic receptors. And I think the last thing we were talking about is how does that work? So how is it that phasic receptors can be phasic? And that was based on this experiment. And it turns out that many of the really phasic receptors that we have, at least for the mechanoreceptors, like Pacinian corpuscles, tend to be phasic because of the structure that they are sort of encased in. So we have the dendritic endings, the receptive endings of the Pacinian corpuscle are held right, within the structure. I'm shaking too much to reveal it, but they're held right in here. But they're encased within these sort of multiple layers of these fibers. And so what this experiment is showing is that when the fibers are present, when you apply the pressure onto this particular receptor, it responds very quickly, but then these layers seem to rearrange around each other, or something's happening with the layers that results in pretty quickly, even though the stimulus is still being applied, the pressure is basically relieved from the actual dendritic endings because of the rearrangement of these layers as they slide past each other. And so the receptor basically stops detecting any kind of a stimulus at this point. It's very quickly ceasing to fire, and it will cease to fire until such time that you actually remove the stimulus, and then we tend to get another burst as we get more rearrangement of those layers when the stimulus is actually removed. But we can negate all of this action here as far as the initial adaptation, and we can also sort of negate this part of it if we remove those layers. And that's what's being shown here. So in this case, if you remove the pressure, or remove the capsule, now if we apply a stimulus onto the dendritic endings, we get a rapid response, and then basically a persistent, effectively a tonic response. This looks like a tonic receptor now. It's not a phasic receptor. It continues to respond and respond and respond and respond over time as long as the stimulus is present. And then finally you take away the stimulus and then you'll drop back down to your sort of standard resting receptor potential. So this is indicative that it's the structure of all these sort of capsular layers around the putative corpuscle that's responsible for the fact that it's a phasic receptor. And the point of that, I guess, is that in a way it's sort of indicating that this property of these things being phasic receptors, like this is a selected trait. This is something where this is clearly very intentional, if you will, in that the structures are designed to function in this way. These putative corpuscles are intended to be phasic. They're intended to stop paying attention to the stimulus very shortly after it appears. And I think the reason for this is that this allows these kinds of phasic receptors to be really good at picking up on new things that are happening. So the first time, is immediately, once something touches your skin, Meissner's corpuscles, which are phasic, and Pacinian corpuscles, which are phasic, they start responding like crazy, and then they stop pretty quickly after that until some new stimulus comes along. And so they're very good at picking up anything that's new. So this is, the little tiny bug that's barely crawling on you or barely touching you and not causing much of a stimulus. You can feel that in large part because you have, that'd probably be Meissner's corpuscles, that are very sensitive to new things happening. Whereas the more tonic receptors, the ones like Merkel discs and like the Ruffini endings, these guys, these are more sort of like the ones that you're always aware of what's happening. So, you put your sock on in the morning and you can immediately feel the sock go around your leg, but then you kind of stop paying attention to it. That's because at this point, your Meissner's corpuscles and your Pacinian corpuscles start ignoring the sock. If you really think about it right now, you can probably still feel the sock on your foot. But it's different from the moment you put it on there. Even though, like if you're like me, you pull the thing down, there's like elastic band marks and stuff on there, so it's clearly been applying pressure. It's just that these phasic receptors just very quickly start to ignore it. And the only receptors at that point that can pay attention to it still would be the ones that are tonic. But it's not changing. It's probably kind of harmless at that point, unless it starts causing you pain or something. Long-term stimuli that aren't really changing often aren't very interesting. And so that's why we have these guys designed to find the most interesting new stimuli, because those are the ones that are most likely to be interesting, relevant, potentially dangerous, that we need to pay attention to. Yeah? That's a great question. Yeah, so is it possible that autism, something's going on with autistic individuals where their phasic receptors aren't either working properly or being received properly? I don't know. I don't even want to speculate. Sure, but I don't know, and I've never heard of anything like that. But it's a great idea, because you do have individuals that seem almost unaware of their surroundings. So is that happening at this receptor level, or is it happening at the brain level? I lean brain level, but I don't know. But do you have a...
Well, just in doing the observation with kids with autism, they always start off the session with little brushes. They brush your skin.
Uh-huh.
Is that desensitizing the nerves?
I don't know that either. That's great. I've never worked with autistic patients, but that's a great... It could be stimulating these different kinds of receptors, and then...
The other thing I wonder is, over time, does their sensitivity to that brush get less and less as the more frequency to it? So the point that they...
Yeah, so what kind of acclimation they have over time. I mean, all of this, I'm gonna speak to this stuff. All of this stuff is at the matter on a time frame of seconds to minutes, as opposed to months of repeated stimulation. I don't even have a good answer for you on some of these ideas on autism, though. Do you have a... Yeah, okay. So diabetic neuropathy, so people are losing sensation in their feet, for example, and then they have, then they start to injure their feet and don't realize it, and they get infections. my understanding of what's happening with diabetic neuropathy is it's not so much at this like immediate mechanoreceptor level. It's more that the axons that are carrying that information up to their brains basically are punking out or starting to die. And so I think it's actually an osmosis problem, which is what I always thought was really cool about it. So, so I'm drawing a picture. So the idea is, as I understand it, at least one of the things going on. So if this is a blood vessel, and then here's a neuron running right next to it, is that the blood has lots of glucose because they don't have insulin or they're not responsive to insulin, and so as a result they don't pull the glucose out of their blood, so they start to have really high glucose levels, which means they have really high solute concentration inside the blood. Water will flow from regions of low solute concentration to high solute concentration, and so the tissues, water fluid starts to flow from tissues into the blood, and that would include from the tissues and the cells that are surrounding them. And so what I think literally can happen is you start to lose fluid, like fluid is exiting axons and getting into the blood, and so you kind of desiccate your neurons, and this leads to death or at least malfunction of the neurons in the area. So this is that's how I understand what's going on with a lot of diabetic neuropathy. So it probably is more like at the axon level as opposed to at the actual receptor. That's a great question, though. I think I had somebody write their neuroscience paper, like when I was writing papers, I'm not doing papers this semester, but the whole thing was on this. It was great. It's a good paper. Other questions? So the fact that you could look for a paper on that if that's a topic that's of interest for your paper summaries. Okay, so this gives us kind of a feel for how mechanoreceptors work. And again, I think the thing that I think is fun about all these different kinds of sensory receptors is that each of them is their own little machines. And so in the case of mechanoreceptors, pretty much in all cases, there's some kind of stimulus that opens little channels on the membranes of these dendritic endings, and that allows ions to start rushing in, and it depolarizes the cell and starts sending action potentials up those axons. So usually these are little channels that are gated that just, when you physically stress the channels, they open. And so as a result, you get a depolarization and action potentials flying up these axons. As they travel up the axons, different kinds of receptors feed into different sorts of axons. We kind of talked a little bit about this a few chapters ago, where axons from the skin will have different classifications based on their structure, depending on what kind of receptors that you're dealing with. So basically, there's four main classifications of axons that are coming out of the skin. They're called either A alpha, A beta, A delta, or C. I don't know why they didn't keep going with the, go to gamma or something, but they just go A alpha, beta, delta, and then C. And these are basically arranged by the size of the axon. So here is indications of diameter of each of these different varieties of axons. So these are varying from 13 to 20 micrometers. These are readily visible in a microscope, down to these guys, which are like 0.2 to 1.5 micrometers. These are getting hard to see. They're getting to be sort of hair-like in structure. So they're varying dramatically. I mean, these largest axons up towards 20 micrometers would be, 100 times the diameter. Diameter, not area, like 100 times the diameter of some of these little tiny class C fibers. And as we've seen, the diameter of the axon has a big effect in terms of the speed of transmission. And so that's actually indicated down here. So large diameter axons have very low internal resistance, as we've talked about. And so as a result, these really large axons have very rapid signal conduction. They're sending signals 80 to 120 meters per second. Whereas these fine little ones are sending signals that are, 0.5 to 2 meters per second. So this is getting down to the point where, 2 meters, we're like, we're maybe 1.5 meters long. So if they're going, let's go in the middle of this range. If a signal is traveling 1 meter per second, that literally means that it could take a good 1.5 seconds to get from your toe up to your brain if it's traveling on these axons. That's getting to be a meaningful delay in some kind of a sensory receipt that you're getting. These different kinds of sensory axons are each associated with different kinds of receptors. So all the mechanoreceptors that we're talking about, they pretty much all are classified in this group here, these A-beta axons. So these are still pretty big. They're not as big as the A-alphas, but they're big. So large axon diameter. And I guess the other thing you can sort of see is that there's also a difference in myelination. So the further you go up in terms of the size spectrum, the more myelination you have, and therefore the more insulation, the more membrane resistance you have, which is also going to increase your action potential propagation velocity. So touch receptors tend to feed into axons that are these A-beta axons, and they tend to travel, send signals very quickly. Other kinds of somatosensory inputs tend to be using some of the smaller fibers. So like, and these are described down here. So pain, temperature reception, itch, these kinds of sensory receptors feed into some of the smaller fibers like the A deltas or the C fibers. And so they're sending that information much more slowly than we're used to receiving. There. Yeah, so we were talking about that, right, a while ago, that, you know, if you, like the next time you get a paper cut, try to think about this as it's happening. Because you oftentimes will instantly feel something happen on your finger, and you're like, oh, that might not be good. And like that, this is always the first thought. And that's where you're receiving that first bit of touch information. There was some abrasion that happened, and so you felt the mechanoreception then, but you don't actually feel pain yet. And this is like a split-second kind of sequence, but this is what goes through my head when I get a paper cut. So that information is coming up these A-beta axons. This is all touch information, right? But then if you in fact did injure yourself, moments after that, a split second after that, you'll start to get the first kind of sharp, twingy kinds of pain that comes up. That's probably coming up, that kind of pain, that first round of pain, is coming up these A delta neurons. So these do contain pain information. Some of the pain receptors go into those. And so then you're kind of getting this, oh, yep, okay, that's a paper cut. And then what'll happen is that maybe literally, you know, a second might have passed since the first stimulus that you felt, but then you start to get pain inputs coming from these guys, the C fibers, and then that's kind of like the dull, achy pain that kind of builds and gets, you know, it was like, oh yeah, okay, that's really definitely a paper cut at that point. So there's sort of like a series of 3 stimulations you get through the process. At least this is what I've perceived, and I try to pay attention whenever I do injure myself this way. A couple of the things that are shown on this chart, and I kind of got out of order with my questions and wordage over here. So this is classification for axons from skin. And in a way, this is a little bit of a theoretical classification. That's not a hand, right? So it's a little bit of a theoretical classification because really these A-alpha axons don't really exist much from skin. There's probably a couple that fall into that category, but for the most part, sensory axons coming out of the skin don't fall in this A-alpha category. They just never really get that big. If they did get into the 13 to 20 micrometer area, we would call them A-alphas. But you can almost put an X through that because we don't really have those. Instead, we basically start with A-betas. That's as big as we get from skin. That's our touch reception, mechanoreception. And then the A-deltas would be like pain and temperature. And then the C's would be more pain, temperature, and then itch falls in this category as well from our itch receptors. If, on the other hand, you're looking at somatosensory information coming from muscles, so this is telling you what your muscles are doing, how fast they're moving, what they're up to, we have an entirely different classification scheme for them. We call them group 1, group 2, group 3, and group 4, and sometimes these are subdivided. So in, I think, one of the next chapters, when we talk spinal cord and spindle apparatus, we'll talk about group 1A receptors and group 1B receptors. So, you know, this is the system for all the things coming out of muscles. Literally what's happening, or what you're seeing here, is the manifestation is that you had, of the fact that you had one group of scientists, and they all talked to each other, and they worked on sensory receptors from the skin, and you had an entirely different group of scientists that all talked to each other, that all worked on sensory receptors coming from muscles, and they didn't talk a lot across each other, and so they ended up kind of reinventing the same classification scheme, but they did so with a different terminology. So again, if it's coming from muscles, it's group 1, 2, 3, or 4 based on these diameter sequences. Whereas if it's from the skin, it's a alpha, beta, delta, or C. But again, for skin, really we're kind of mostly in this range, beta, delta, or C. Coming out of muscles, at least for the ones that we're going to talk about, we're pretty much all in this group 1 category. And very few of the axons that are sensory receptors that are coming out of muscles, telling us what our muscles are doing, are even down into group 2. They tend to be very, very large. And they're going to power some of the muscle reflexes that we'll talk about when we talk in the spinal cord chapter. Any questions on that stuff? Makes sense? I always think it's funny when you've got things like that happen. It's like just a historical accident. If we were going to do it, like somebody at some point, I just feel like at some point we're going to reconcile that and just everybody's going to have to agree. But that means one of those groups is probably going to have to agree to follow the other group, and they probably don't want to because that's what the other group does. And they want to be right. Okay. This is also a reason that, you know, certain structures have like 5 different names. It's the way they do it in neuro. Okay, so at this point, we've got a little sensory receptor somewhere, a mechanoreceptor we're focusing on for the most part, and we've talked about the axons that are sending those signals, so we need to start following the signals now out of our peripheral nervous system and get into the central nervous system and then start following where those go within the central nervous system. So that's kind of like the focus of the rest of the chapter is where do those axons go that originated out in our skin, sending touch information in, where is that, what's the pathway by which that travels through the central nervous system? So all somatosensory information is ultimately going to be carried to the central nervous system by sensory axons, and they'll be carried in sensory nerves. So these nerves are bundles of axons, and so we'll have nerves that'll service particular swaths of skin, and they're receiving axons from all the sensory receptors in that swath of skin, and then ultimately channeling that to spinal nerves. And so this is where we're getting back into the chapter 7 stuff, the anatomy stuff we just talked about. So here's a little Pacinian corpuscle, and we're sort of following that all the way in to spinal nerves, and we have basically, as you move along the spinal cord, we've got a spinal nerve for each segment along the spinal cord. All the sensory neurons that are coming in through these sensory axons that fuse with spinal nerves ultimately will go in through the dorsal root of the spinal nerve. So we kind of pass into the vertebral column here through the spinal nerve. So the edge of the vertebra is here, and then it kind of wraps around this whole structure. So we're going into the vertebral column here. We go through this structure called the dorsal root ganglion, and then ultimately the axon travels into the spinal cord, arriving here in the dorsal horn of the gray matter. One thing you can sort of see, so this red line here with the little dot, that's supposed to indicate the sensory neuron that's bringing this information in. Can you just sort of tell a diagram from that little diagram what kind of neuron this is? Do you remember, like, these are neuron classifications based on structure from back in chapter 2. So we've got, this is the axon. And then the cell body is really what this is supposed to be here. So what kind of axon is this? Or what kind of neuron is this? Do you remember? This is like a long time ago now. Something polar. No? Yeah, it's unipolar. So the cell body, the polar thing basically refers to how many processes are coming off there, how many dendrites and axons are coming off that one cell body. And in this case, these kinds of sensory receptors, they're all feeding into unipolar axons where there's just, the cell body is kind of marginalized off to the side in here, and then it's connected almost like just sort of as an afterthought to the axon, which just totally bypasses it. The axon is starting over here with the receptive endings, and then it feeds all the way through here, and it just kind of drives right by the cell body, almost like it doesn't matter. And the cell body, of course, is providing all the protein and mitochondria and such and ATP that are allowing that axon to function. All of those cell bodies, in fact, for all these unipolar axons are collected in this structure, this dorsal root ganglion. So the reason we have a dorsal root ganglion is that this is the place where we house all the cell bodies for all these sensory axons. And again, their axons may start way over, like on the tip of your finger, or on the tip of your toe, but their cell body for those cells is always in this dorsal root ganglion, wherever they're coming from. And all the somatosensory neurons, for the most part, out in the peripheral nervous system, originated as these kinds of unipolar neurons that have this dorsal root ganglion cell body. I feel like there was another point I wanted to make, but I can't remember it. Okay, so axons are coming in, and you can see we're just sort of drawing an arrow here because we need to figure out what happens at that point. Before we get into like what happens inside the spinal cord, I do just want to give kind of an overview of spinal cord overall. So again, here's your brain. Here's the spinal cord going down. The spinal cord basically ends as you sort of like this is trying to show the vertebra that are nearby. So these are up here. These would be thoracic vertebrae, which are the ones that are kind of in this area, and then the lumbar vertebrae are in that like that small of your back that you can kind of, there's sort of a notch so you can sort of push in this area. This is the lumbar vertebra, and the spinal cord basically ends right at the top of that. So like right around here, this is L1 vertebra, and this is the end of the spinal cord right here. But what you can sort of make out, and it's easier to see it over here, is that even though the spinal cord basically ends, it gives rise to lots of spinal nerves at this area, and many of the spinal nerves continue down the vertebral column. So all down through here, there's this structure. It's called the cauda equina, which literally means horse's tail, because it looks a little bit like a horse's tail when viewed from behind. And that fills up the rest of the vertebral column, going all the way down through the lumbar vertebra into the sacral vertebra. But the spinal cord itself ends basically at the top of the lumbar vertebra. All the way down along the way, you've got a series of spinal nerves that come in. So they're always named for an adjacent vertebra. So we have 8 cervical spinal nerves. There's technically only 7 cervical vertebra, but we start naming them for the first one, and then we kind of go past the last cervical vertebra. So there's 8 cervical spinal nerves, and there's 12 thoracic spinal nerves. So that's each of these. There are 5 lumbar, and then 5 sacral spinal nerves. I think there's some coccygeal ones too, but it's not really showing those. Each of them, though, it's basically a big segmented structure as you're moving your way down. And each one is receiving sensory information from different parts of skin that are associated or basically peripheral or adjacent to where these spinal nerves are coming in. There's also motor information going out all these spinal nerves. So we have axons coming out of our spinal cord that are traveling to skeletal muscle that are coming out through each of these spinal nerves, and then they go on their destination to the particular muscles that they're going to be stimulating. You can actually see where the spinal nerves are coming in on this picture. So this is a picture that's showing all these different patches. They're called dermatomes of skin that service each of those different spinal nerves. So for example, let's do this one. So this guy, he's got this patch right here, which is kind of like right here across his chest. This patch is called the C5 dermatome. And the C5 dermatome would feed in primarily to the C5 cervical nerve, which is right up there in his neck. If you go just a little bit above the C5 space up here, you get into a C4 space, and then a C3, and then a C2. So the idea is that each of those patches of skin is feeding primarily into one particular spinal nerve. So I'm going to go back a slide. So here's C5. So most of that whole patch of skin is feeding into this spinal nerve, which is coming in at this level of the spinal cord. But again, if you go to a little bit higher patch of skin, you'll feed into C4. Or a little bit higher than that, you feed into the C3 spinal nerve, which is named after the, associated with the C3 dermatome. You can see that basically works all the way down through the torso. It gets a little weird when you get into the arms, and I always sort of was, it always kind of struck me of like, well, why are we changing direction in the arms? And I guess if he held his arms out to the side, it might work, but the same thing happens in the legs. We go from kind of like these horizontal dermatomes to these vertical dermatomes. It never really quite made sense to me as far as why that was until I saw this picture in your book, and this kind of explains it. We're not descended from upright organisms. We're descended from on 4 legs organisms. And so if you have a person bend over, touch their toes, what you see is that actually these dermatomes are perfectly horizontal. It's just they head straight down, you know, each of the arms and each of the legs. So it lines up actually really, really, really nicely. So these are dermatomes themselves. I mean, again, the idea is that each patch of skin feeds into primarily one spinal nerve. It's not as clean as we're really illustrating it. So this C5 patch, again, it definitely sends lots of axons into the C5 spinal nerve, but it also sends axons into the C4 and C6. It's just not sending it down to like T9 is the point. So for the most, there's always a little bit of overlap. Any one dermatome will actually be talking to several different spinal nerves and sending axons to several different spinal nerves. But it's still pretty localized. So clinically, this can be useful if you have a patient that comes in and has some kind of a spinal cord injury, and you're interested in trying to localize where is that spinal cord injury. And you can kind of go down the person's dermatomes if you have some landmarks, which we do, and basically identify, you know, maybe they can feel down to here, but they can't feel the next level down. That would indicate that, you know, maybe they've got, they're okay at T9, but maybe there's damage around the T10 spinal nerve or the T10 level of the spinal cord, and as a result, they're not really able to feel much past that point. But above that point, they may be able to perceive just fine. So they can be useful in a clinical sense as well. Yep. Yeah, so what would happen if one of these spinal nerves is partially severed? I mean, certainly any axons that are cut in the partial severing are going to be non-functional anymore at that point. What often will happen when you have any kind of nerve damage or spinal cord damage is that you also then have a lot of inflammation that happens, especially over the first several days. And so you may have, you know, a sort of an immediate level loss of function, and then that actually may progress and get even worse over some time because the inflammation comes in, and as inflammation comes in there, it disrupts the function of any additional axons that are in that area. If it's peripheral nervous system, so if it really is spinal nerve, there's a chance that there could be regeneration and healing of those axons. Unfortunately, if it's spinal cord, there are literally chemical signals that inhibit regrowth, and it's very frustrating. And so as a result, it's basically not healable in spinal cord injuries. So one of the interesting lines of evidence, and we almost always read a paper about this in Neurosem at least every couple of years, is efforts to inactivate some of the signals that inhibit regeneration in the spinal cord, because if you can do that, then all of a sudden you're able to start healing spinal cord injuries, at least a bit, you know, and that would be very nice to be able to do. Question? Yeah. 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 So the reason the face is not shown up here is that this is pretty much all trigeminal nerve. There's a couple other sensory cranial nerves, but most of the face proper is all feeding into the trigeminal nerve. So yeah, good question. Yep. I don't really know. It doesn't seem to be labeled up there, and I don't have a good answer for you. I also don't know why we stop at C5. So, yeah. I've been meaning to look that up, and I haven't. Oh, yeah, you're right. Okay, yeah. 6, 7, 8's down here. All right, good. So that's explained. That's not labeled on that one, but whoops. Yeah, okay. So now at least we've got down to C8, but I don't know where C1 went. C1's a little weird. It's emerging above the level of the first. It's just kind of weird in that it does emerge between the cranium and the first vertebra. So this first vertebra is that weird, if you've had anatomy physiology, it's the atlas. So it's kind of an odd area anyway, but I don't know. I don't know what it's doing if it's not getting any sensory input. So, I mean, I guess it could be motor. Other questions? These are great. Excuse me. Okay. So now that we kind of know where they're getting in, we understand this is happening at every segment. So let's basically try to follow an axon as it goes into some level of the spinal cord. Doesn't really matter which level. They're all kind of doing the same thing. And then follow that up into the brain. So we're going to follow mechanoreception axons, which would be mostly, almost entirely those A-beta classification axons. So these are pretty big, pretty heavily myelinated, sending signals pretty darn quickly, and they're traveling up into the spinal cord, as we said. They go up through the spinal nerve, they go up through the dorsal root of the spinal nerve, and they come in through these rootlets, and then we need to figure out like what happens to them. The main flow of information, as I kind of think about it, is that it actually, at this point, just completely bypasses the gray matter of the spinal cord. So here's this axon, and you can see it's sort of immediately going not into the gray matter here, per se, but it's immediately going into the dorsal columns. So this is all white matter, and it's then continuing up the axons, or up the white matter toward the brain. So we have, little touch receptors at the ends of our fingers or the ends of our toes that are getting this touch information. They're coming all the way to the spinal cord, which is already a long distance for a little cell process to go. And they're still not ending. They're just taking a U-turn and now going straight up the spinal column toward the medulla. And they still haven't really ended. That's still all the same axon, which is just blowing my mind. So, that's all the way from the tip of your toe. Now we're going into the spinal cord, and now we're going all the way up the spinal cord through these dorsal columns. I think as we mentioned last time, the dorsal columns of the spinal cord are almost entirely all sensory information. And most of it is this kind of touch sensory information. And so this is one of our major pathways by which and really the major pathway by which touch information goes up our spinal cord toward our brain. There are, however, as you can see, some synapses. So this is like an axon collateral that heads toward the brain. But there's other collaterals that kind of split off and do go into the gray matter at the spinal cord. They're going to enter into the dorsal horn of the gray matter, and they'll form synapses with axons that are in that area that may trigger various reflex arcs. So they are feeding into some of the spinal cord circuits in this area. One thing we'll see later, probably not today, but in maybe Monday, is that some of these touch receptive inputs feed into and interact with how pain receptive inputs work. So as pain is coming in, the pain is kind of modulated by touch. And so that's what some of these axon endings are doing, is that they're going to be coming in, interacting with neurons that are in this area that are getting other kinds of sensory information coming in. For the most part, though, we have axons, like the most important line of signal is coming in through dorsal root, going in directly into the dorsal column, and then we're traveling up, it says right here, up the ipsilateral dorsal column of white matter to the brain. Ipsilateral, ipsa just means same, so this is the same side as. So that means that if this is a signal coming from the right, or from your left hand, I'm holding up my left hand, and coming into the left side of your spinal cord, it is staying on the left side. It's not crossing over at this point. So we are staying on the left side all the way up into the brain. So this big neuron that's carrying all this information, all this distance from your toe or from your finger or from any other spot in your skin, up into the spinal cord, up into the dorsal column, all the way up towards your brain, we call that one the primary or first order neuron. That's down here. It eventually is going to form a synapse, but it's really not going to form a synapse till you get all the way up into the medulla of the brain. At that point, it'll give rise to a second order neuron that's receiving that information from the primary or first order neuron and then sending that information on further. But it's those first order neurons are carrying information all the way from the skin all the way up into the brain. So because it's coming up these dorsal columns, the name of the pathway, or one of the names that you'll see for the pathway that's providing all this, or that's carrying all this information, is what's called the dorsal column medium lemniscal pathway. And so this is this overall pathway by which touch information is traveling up your spinal cord and then ultimately through your brain. Excuse me. We're going to see a lot of this kind of a figure in this book, in this chapter, as well as in the chapter on motor control, spinal cord motor control. And what they're trying to do with this is sort of represent all the different structures that this signal is traveling through on its way up to the cerebral cortex. And they do so kind of in cross section. So we sort of see these in series. So first they're showing you a cross section through the spinal cord. So that's number one. That's down here. So here's your cross-section through the spinal cord. This is basically the same thing we saw on the prior slide. I guess they swapped one side for the other, but again, we're coming in through the spinal nerve. We go up through the dorsal root. We have some synapses that are going directly in the gray horn, but the main line of information is going directly into the dorsal column. And then we'll travel up the dorsal column all the way up the spinal cord until you reach the medulla, which is where their next slice is. So here's the slice through our medulla. And in this case, they're showing a collection of dorsal column nuclei that are up here. So these are sort of in the dorsal part of the medulla. There's a collection of nuclei. So these are collections of gray matter in the medulla that are receiving all that information. So those first order neurons are feeding into these two nuclei. There's two of them on each side. So there's these two, and then there's these two. I think at some point I do. Yeah, I'll give you some names for some alternative names for these in a second. But again, the idea is that they're feeding into here. So your first order neuron comes to this point, and then is going to form synapses with second order neurons. And it's not even really like a one-to-one kind of a synapse. What's happening is you've actually got a bunch of axons that come in, and then several of those axons will all sort of feed into the same second order neuron, which kind of pulls together all their information. There's a little bit of an interpretation period. But then, assuming it's getting adequate signal, it will then send a signal up the second order neuron, which travels from the medulla up toward the thalamus, which is our next stop. One thing you can see, though, is that immediately this second order neuron doesn't stay on the same side of the central of the nervous system. So we came in this case, we're coming in on the right side, based on how I'm standing here. I guess that's your left, so let's say left. So we're coming in on the left side, and we went up the ipsilateral side of the dorsal column. So we're still on the left side of the medulla when we form our synapse in the dorsal column nuclei. But then the second order neuron immediately decussates. So it's crossing from the left side over to the right side and then traveling up this structure called the medial lemniscus, which is sort of this band of white matter at the heart, or that's very deep inside the medulla. So we call this thing the dorsal column medium lemniscal pathway because we start in the dorsal column and then we switch over and travel up the medial lemniscus, which basically takes us all the way up to the thalamus, where we have our next synapse. In the thalamus, so now we're over on the right side of the thalamus, we then have another nucleus. It's called the ventral posterior nucleus. So that term is right there. Here they're abbreviating it VP. That's the ventral posterior nucleus of the thalamus. We get another synapse between the second order neuron and the third order neuron. And again, it's usually like there's several second order neurons that are feeding into each third order neuron. But the third order neurons then finally will take the information from the thalamus up to the primary somatosensory cortex, which is the first place in the cerebral cortex where we ever receive that information. In the last chapter, we were talking about the thalamus as being like the gateway to the cerebral cortex. So all sensory information goes through the thalamus before it gets to the cortex. So this right here, that's this sort of in action. So here we've got the thalamus receiving information, probably doing some filtering, and then relaying that information up to your primary somatosensory cortex. So the next slide kind of summarizes all of that, so I'm just going to talk it out. So we had A-beta axons, so you should be able to understand all the stuff that's on here. So A-beta axons entered via the spinal nerve. They branched. Some of them synapsed in the dorsal horn of the gray matter, but others went up the dorsal column. Your primary afferents are just incoming neurons. So these primary first order neurons synapse in the dorsal column nuclei of the medulla. And they give rise to second order neurons that decussate, so they cross from one side to the next. And then they travel up the medial lemniscus through the brainstem up to the thalamus, and they ultimately form a synapse in the ventral posterior nucleus of the thalamus. And at that point, they give rise to third order neurons that take that information all the way to the primary somatosensory cortex of the parietal lobe. Each step along the way, going from first order to second order and second order to third order neurons, we're consolidating information. We're modifying the information a bit. There's opportunity for signals coming down from the brain to kind of suppress or amplify the input. But ultimately then, it's carried to the primary somatosensory cortex. And at that point, it gets to our cerebral cortex, and we might start to have some conscious perception of that touch information. If you took human anatomy and physiology, I'm sorry, I'm just going to say this one thing. If you took human A&P, You probably didn't call it the dorsal column medial lemniscal pathway. You probably called it two things. You probably called it the gracile fasciculus and the cuneate fasciculus. It's the same thing. Basically, we talked about there being like two sets. So there's actually two parts of each dorsal column. There's two dorsal column nuclei. And these are associated on each side. And these are associated with the gracile and the cuneate fasciculus. The idea is that this one is servicing all the spinal nerves T6 and below. So, you know, kind of take a line here. Everything that's coming in from below that point. So your lower legs, your lower abdomen, they're coming in from through the gracile fasciculus. If it comes in from your upper body, it goes in through the cuneate fasciculus. They're all doing the same thing. They're just sort of running side by side each other as they're traveling through the spinal cord. So we were ipsilateral here. At this point, we have gone contralateral once we decussate. All right, I'm out of time, so I will see you guys Monday. Have a great weekend. See you then.